As we have seen, the invention of recombinant DNA methods in the early 1970s resulted in a round of controversy and soul-searching centered on the Asilomar Conference. Now it is happening all over again. At the time of Asilomar, it may at least be said, we were facing several major unknowns. We could not then say for certain that manipulating the genetic makeup of the human gut bacterium E. coli would not result in new strains of disease-causing bacteria. But our quest to understand and our pursuit of potential for good proceeded, however haltingly. In the case of the present controversy, anxieties persist despite our much greater understanding of what we are actually doing. While a considerable proportion of Asilomar's participants urged caution, today one would be hard-pressed to find a scientist opposed in principle to GM foods. Recognizing the power of GM technologies to benefit both our species and the natural world, even the renowned environmentalist E.O. Wilson has endorsed them. Where genetically engineered crop strains prove nutritionally and environmentally safe upon careful research and regulation, they should be employed. The opposition to GM foods is largely a socio-political movement, whose arguments, though couched in the language of science, are typically unscientific. Indeed, some of the anti-GM pseudoscience propagated by the media, whether in the interests of sensationalism or out of misguided but well-intentioned concern, would be actually amusing were it not evident that such gibberish is in fact an effective weapon in the propaganda war. Monsanto's Rob Horsch has had his fair share of run-ins with protesters. I was once accused of bribing farmers by an activist at a press conference in Washington, D.C. I asked what they meant. The activist answered that by giving farmers a better performing product at a cheaper price, those farmers profited from using our products. I just looked at them with my mouth hanging open. Let me be utterly plain in stating my belief that it is nothing less than an absurdity to deprive ourselves of the benefits of GM foods by demonizing them. And with the need for them so great in the developing world, it is nothing less than a crime to be governed by the irrational suppositions of Prince Charles and others. A few years from now, when the West inevitably regains its senses and throws off the shackles of Luddite paranoia, it may find itself seriously lagging in agricultural technology. Food production in Europe and the United States will come to be more expensive and less efficient than elsewhere in the world. Meanwhile, countries like China, which can ill afford to entertain illogical misgivings, will forge ahead. The Chinese attitude is entirely pragmatic, with 23% of the world's population, but only 7% of its arable land. China needs the increased yields and added nutritional value of GM crops if it is to feed its population. On reflection, we erred too much on the side of caution at Asilomar, quailing before unquantified, indeed unquantifiable, concerns about unknown and unforeseeable perils. But after a needless and costly delay, we resumed our pursuit of science's highest moral obligation, to apply what is known for the greatest possible benefit of humankind. In the current controversy, our society delays in sanctimonious ignorance. We would do well to remember how much is at stake: the health of hungry people and the preservation of our most precious legacy, the environment. In July 2000, anti-GM food protesters vandalized a field of experimental corn at Cold Spring Harbor Lab. In fact, there were no GM plants in the field. All the Vandals managed to destroy was 2 years' hard work on the part of 2 young scientists at the lab. But the story is instructive all the same. At a time in which the destruction of GM crops has become positively fashionable in parts of Europe, when even the pursuit of knowledge on that continent and this one can come under attack, those in the vanguard of the cause might do well to ask themselves, what are we fighting for? Chapter 7. The Human Genome. Life's Screenplay. The human body is bewilderingly complex. Traditionally, biologists have focused on one small part and tried to understand it in detail. This basic approach did not change with the advent of molecular biology. Scientists, for the most part, still specialize on one gene or on the genes involved in one biochemical pathway. But the parts of any machine do not operate independently. If I were to study the carburetor of my car engine, even in exquisite detail, I would still have no idea about the overall function of the engine, much less the entire car. To understand what an engine is for and how it works, I'd need to study the whole thing. I'd need to place the carburetor in context as one functioning part among many. The same is true of genes. To understand the genetic processes underpinning life, we need more than a detailed knowledge of particular genes or pathways. We need to place that knowledge in the context of the entire system, the genome. The genome is the entire set of genetic instructions in the nucleus of every cell. In fact, each cell contains 2 genomes, one derived from each parent. The 2 copies of each chromosome we inherit furnish us with 2 copies of each gene, and, therefore, 2 copies of the genome. Genome sizes vary from species to species. From measurements of the amount of DNA in a single cell, we have been able to estimate that the human genome, half the DNA contents of a single nucleus, contains some 3.1 billion base pairs. 3,100,000,000 A's, T's, G's, and C's. Genes figure in our every success and woe, even the ultimate one. They are implicated to some extent in all causes of mortality except accidents. In the most obvious cases, diseases like cystic fibrosis and Tay-Sachs are caused directly by mutations. But there are many other genes whose work is just as deadly, if more oblique, influencing our susceptibility to common killers like cancer and heart disease, both of which may run in families. Even our response to infectious diseases like measles and the common cold has a genetic component, since the immune system is governed by our DNA. And aging is largely a genetic phenomenon as well. The effects we associate with getting older are to some extent a reflection of the lifelong accumulation of mutations in our genes. Thus, if we are to understand fully and ultimately come to grips with these life-or-death genetic factors, we must have a complete inventory of all the genetic players in the human body. Above all, the human genome contains the key to our humanity. The freshly fertilized egg of a human and that of a chimpanzee are, superficially at least, indistinguishable. But one contains the human genome and the other the chimp genome. In each, it is the DNA that oversees the extraordinary transformation from a relatively simple single cell to the stunningly complex adult of the species, comprised in the human instance of 100 trillion cells. But only the chimp genome can make a chimp, and only the human genome a human. The human genome is the great set of assembly instructions that governs the development of every one of us. Human nature itself is inscribed in that book. Understanding what is at stake, one might imagine that to champion a project seeking to sequence all the human genome's DNA would be no more controversial than sticking up for Mom and apple pie. Who in his right mind would object? In the mid-1980s, however, when the possibility of sequencing the genome was first discussed, this was viewed by some as a decidedly dubious idea. To others it simply seemed too preposterously ambitious. It was like suggesting to a Victorian balloonist that we attempt to put a man on the moon. It was a telescope, of all things, that inadvertently helped inaugurate the Human Genome Project, HGP.